I guess I will start just one quick sentence. So Professor Azizi from Florida State University at Tallahassee, Florida, we're going to show us how to do mathematical modeling with applications in epidemiology and pharmacology. Thank you.
Thank you so much. I'm not a professor. Hopefully one day I'm a postdoc. So I'm going to share my screen.
Doctor, it is.
Thank you so much for the introduction. So I hope everyone can see my slides. And please feel free to interrupt me any part of this that you want me to explain more. Actually, I selected this topic. I was talking to Dr. Winkel that this topic actually was the topic I was mentoring one undergraduate student. She was really interested to know about the application of mathematics in like medicine and especially like pharmacology. Also, I will talk about epidemiology. So before getting us started, I would like to acknowledge Dr. Winkel and all organizer committee for such an amazing and informative conference, I would say. And this is online and so many people are attending. I'm so excited to be here. And I'm going to start now. So the topic for my presentation is about how we can model, or sorry, maybe I just close my emails. This is better. They're going to bombard me lots of emails. So that's better, I think. So sorry. So the topic is about how we can use mathematical modeling in epidemiology and pharmacology. For me, it was a very new topic when it was during my PhD, and I was working with a group of like interdisciplinary people in pharmacology, like scientists and many people like chemists, and I was working with them. I was the only mathematician there. So it was really, I would say it was stressful because my language was like mathematician. And it took for me 1 month to fix my language to be something more understandable for people who are who they don't have the same background as my background. So this is a big challenge, I would say, at the beginning when you are working somewhere which they are not mathematician. But yeah, we do a lot as a mathematician. So let's just start. This mathematical and statistical modeling are going to help us to understand all the interactions that happen between different, I'm going to divide my system into different compartments or components. They are going to help us to understand the interaction, how they interact with each other in biology or any other field. For me, is specifically biology. And because we really want to predict the future of the system and the model that we have, we don't want to do a lot of experiments. So we did actually for this project, for one of them, we did experiment on, for example, animal and swine to get the data. This is expensive, but our role as a mathematician is there to find out if we can give them a model, they can use it for prediction for the future. So they don't need to sacrifice animal, and it's also like, it's not expensive, like it's going to be better economically. So this is our, I would say, important role in industry or like pharmaceutical industries, that people, they want mathematician, they need. And basically, when we are building a mathematical model or a computational, for example, model, I would say, we need to definitely have like different experiments to check the model with data, if our model is going to work well or not. And using like different tests, we are going to check that if our model is really a good model for data or not. So maybe we need to change something. And also using the data, we can find the parameters that we have in the model, which actually the like in these industries, people that are looking for these parameters, and we need to optimize them and to have a really right model for a specific, for example, project. For example, what my application was, I want to understand the process of how drug is going to distribute in entire body organs. So we need to develop a model which really completely can cover all of the experimental data that we have. And these models that are going to transform all of the biological like information about the system that we have. So our actually role is transforming all of this biological information about the system into a system of, for example, ordinary differential equations, or for my case is ODE, or sometimes, for example, people who are doing cancer research, or like this tumor cancer modeling, or many other stuff that are using partial differential equations, and there are many other type of modeling. So we transform all of this biological information into a system of ODE to do more analysis that, and we give them, yes, you can use this model for prediction of like future of this system, what is going to happen. And this model is actually, the good thing is when we give them a mathematical model, this is a good thing is they are really flexible for analysis and updates. We can update, we can modify the model and find, for example, new results. So this is really important in this type industries, having a mathematician there. So the basic, I would say the foundation of all of this modeling in pharmaceutical like industries, all of this like drug distribution modeling is it's very simple. It's very simple, just a compartmental modeling. When I was explaining to my student, she was really surprised that we can just, with a simple system of OD, we can model the process of this drug, how when we inject the drug through like different like way of drug administration, what is going to happen for this drug or any nanoparticles or anything that you are injecting to like through body or from different ways, like through food or any other way. It's a very simple model, and the approach is, as I said, compartmental modeling. So what are this compartmental modeling actually? So this compartmental modeling, they describe what is going to happen, as I said, for a drug in our body. And what they do is they divide our body into different, for example, compartments. We have different organs, and this is the reaction of a drug in different organs is different. Maybe it's going to affect kidney or lung, or this is really different. So we are really considering all of these details, and this is actually for the entire body is very hard. And we are assuming, so there are some assumptions. We are assuming that a compartment involves several organs or tissue which are kinetically homogeneous. Of course, in reality, for example, when we are modeling, for example, some tissue, when we are really want to make it more realistic, we know that, for example, for tumor cell growth, we have multiple, like we have heterogeneity, we have multi-time scale modeling, and the approach is going to be totally different. But for these two projects, we assume that the compartments that we have, they're homogeneous. We don't have any heterogeneous like drug distribution. So this is our assumption. And also we are assuming that different compartments, they don't have any like direct anatomical or physiological signification. So in compartmental, in this compartmental modeling methods, we assume that the drug concentration is changing like over time, and we are actually, our goal is finding, they are asking us, for example, they hire us as a mathematician to find this drug concentration in different organs, to find, to estimate this concentration, for example, of different drug in kidney, liver, lung, or any other organs. And we need to also estimate the like parameters for them as well. So these are two important jobs. I'm going to go over that. And as I said, so for this type modeling, we usually use this deterministic approach, this differential equations, but it's there are many stochastic or random approach framework that the problem is going to be harder, but they are more realistic. So I'm going to go over two different projects, maybe I go a little bit faster. So about the infectious disease, and which this is something that very common discussion these days, like after COVID came. So recently, due to the fast spread of pandemic disease, this mathematical, people understand really the importance of mathematical modeling and role of mathematician in epidemiology. And many mathematical models have been developed to describe, based on different type of this infectious disease that we have, to describe this type communicable disease. And these models actually, they describe the mechanism of infectious disease, as well as also they are helpful to analysis the effect of, for example, public health interventions, or all of these control strategies that we are using for controlling disease. For example, like vaccine. I'm going to show actually this time talk about just vaccine, how we control disease using vaccine, and how we can mathematically describe that. In mathematical perspective, actually, we describe biological systems by converting them into mathematical and theoretical framework, and we keep the biological parameters, we keep the same, and we use some like simulation to analyze the model and system that we have, like computationally. And for example, to predict the future of infectious disease, one needs to like study the behavior of each individual, which is really important to understand the behavior of like infectious disease overall. So we need to look at this. So the foundation of or a basic of all of this disease modeling is SIR or SCIR modeling. It, as I said, depends on different type of disease that we have. For example, SIR is the simplest one. As you see in the right-hand side, this is we call it a compartmental modeling. We have different compartments. We have compartment S, means that there are susceptible people who could potentially catch the disease. We have I, which are like people who got like, who are like infected now, the infected people who got the virus. And we have R, which is recovered. So, and there are some, this beta, gamma, this delta parameters, and using all of this, we easily write using the compartmental modeling approach, we can write the system of equation. You can make it based on different assumptions that you have related to the disease. You can write really more complicated models for depends on, as I said, depends on the type of disease. So as you see here, for just general, as we have S is a population of susceptible is going down, and we have infected, and we have like recovered in this simulation when you want to see the solution of the system, for example, or SCIR model. For another type of model, we may have exposed people, people who are infected but they are not yet infectious. I think for cold we don't use this SCIR model. It depends on like different like type disease. So as you see, I have a new compartment and write down the system of equation based on what is going on in inside of the compartment, and based on all of this, easily we can write the equations. And this is a numerical simulation of this model. So what my goal for this one, I work with my student actually, it was to see how we can mathematically explain that the vaccine can control disease. And what we do, mathematicians, they do, like our tool is using optimal control theory. So using optimal control theory, we are going to find out the impact of vaccination strategies. So we assume that, for example, I'm going to start from SIR model that I have. I assume that my vaccine is U. So I'm going to assume this vaccination process by a new, for example, a parameter here. I'm going to apply vaccine on the population of the first or susceptible X1, as you see here. So it depends. I mean, I applied it on because we vaccine susceptible people, right? So you applied on the model that you have, and this is the framework that we use using this type modeling is like in the area of like optimal control theory. People, they do that. So we are following all of this optimization method approach to write the model with considering the effect of vaccine. And then if you look at the right hand side, it's showing the, for example, effect of vaccine on the how it works here. We have some at the beginning, for example, if this is a population of infected, using this control, when I plot this solution, or the population size, actually each one is a population size, is going down. There is a little bit going up. It should be some other condition, but as overall, when we apply, for example, vaccine on the population of susceptible, as you see, infected is going down. This is what we want to have it, and susceptible is going up after applying this vaccine. Or I am going to apply vaccine on a Scir model. Again, the same approach, using optimal control theory, writing down the objective function, writing the system of equation using Scir modeling, and see, for example, if I apply vaccine here, what is going to happen for the population of, for example, infected, what I'm looking for, which is decreasing. This is what we are really expecting. For example, vaccine is going to control disease. So maybe someone can use other approaches for modeling and see the effect of, for example, mask or any other control strategies that people we are now actually applying during our daily, for example, life, and write the model and see the effect of really this, for example, control strategies to control spread of disease. And what are other analysis that we can do on a model is the approaches. We want to see the effect of this changing the parameters, all of the parameters at the same time. We want to change these parameters, which are really important for us, and see what is going to happen for the output of the model, which is, for example, here, population size, population of susceptible, infected, or other, for example, compartments. And what our tool here is global sensitivity analysis. There are so many algorithms, but here I'm going to just apply one of them. So what does it do actually? So this analysis, this type analysis allows us to, so global sensitivity analysis, in this analysis, we are changing all of the parameters at the same time and over entire range of interval. If you, for example, apply only local sensitivity analysis, it's just you are changing one parameter, which is not like realistic. It's not good. We want to see that when we are changing all of the parameters, what is going to happen for the population of, for example, susceptible. It's important for us. So it's a little bit harder, but I mean, this is more realistic in this approach. So we are going to actually see in this type analysis, we are going to evaluate the relative effects of each input parameters, and we are going to identify the interaction between the parameters. So that's why we run this analysis. In this analysis, we determine that with variation of input parameters in a certain range that we have, which parameters they have most influential impact on the output, on the behavior of the model. So this is the goal. What I'm using here is for this global sensitivity analysis is a Latin hypercube sampling method, LHS method. So what people have, there are many different approaches people use. I use this sampling approaches. So it's very simple and very fast. This is a sampling method LHS and requires fewer sample actually to compare compared to the other sampling approaches that we use to get the same actually accuracy. And in this method, actually, we divide the random parameter intervals distribution into n equal probability intervals. We are going to finally get a matrix of, so we have n probability intervals, which is our the random parameter distributions, and we have k plus 1, which k is the number of parameters which we are choosing to vary them, change them. So for the case that the interval of the variation for some parameters is very large, then we need to do a like form approximation. So in this method also, sampling is independent for each parameter that we have. For example, for Scir, I think I have 4 parameters. I'm going to change them. I'm going to see, so it's independent, it's really independent for each parameter and can be done by, we're going to do like randomly, like sampling, and we select the values from each probability distribution function. And we also, so these are the assumptions, we sample each interval once for each parameter without any replacement. This is the like assumption. So, and finally we get a matrix of n rows, right? And k, which is the number of parameters, columns of the number of k is the number of parameters that we are changing. So we have n model solutions, we have k parameters, we get this LHS matrix, and we get the output with change when we are changing the parameters each one in the range that we have. And this is the result of, this is for SIR model. We had 3 parameters for SIR. For example, as you see, some of the parameters like gamma is positively is going to affect the model output, some of them negatively. So these are something that for people who are really familiar with this type, with disease, and they can analyze it better and give us like the result. Or for example, SIR, we had like for 5 parameters, and again, some of them positively, some of them negatively affect the population of, for example, susceptible, exposed, infected, and recovered. And what I'm gonna go over other project. Maybe I finish it and then ask like if anyone has any comment, because this project is very interesting. Actually, it was for me really interesting about the mathematical modeling of drug distribution throughout body. I'm going to start with a very simple case, just with 3 compartments. As I said, our approach here is compartmental modeling. So I'm going to start with this, just 3 compartmental model, and then I'm going to develop it to the human, like a body, entire human, like body. So this pharmacokinetics here that I'm putting here, this name here, refers to the rate and extent of distribution of a drug to different tissues. So we are looking for this, right? And also the rate of elimination of the drugs. And this pharmacokinetics, so they call it actually pharmacokinetic modeling. Sometimes they call it compartmental modeling, different like literatures. This pharmacokinetics can be reduced to mathematical equations. This is what we do as a mathematician there, which describe the transit of drug throughout the body. So we have like absorption of drug, and then distribution, and then metabolism and excretion. So this is a very simple like model. I put it here to be more understandable here. As you see, we have this drug or nanoparticles is coming from arterial, which is, I should maybe put a very scientific picture, but this is, I started like from very like in detailed picture, and then I draw this for myself, because based on this, I'm going to find a schematic diagram and then write the model equation. This is how we do in this type like modeling. So I have this like picture of what will happen, for example, for this nanoparticles or drug. Drug is coming from arterial and then going to capillary and then going to deep like tissue. Target tissue can be a tumor cell or any tissue that we are targeting our medicine, drug, anything that we have. And then some of the like remaining of this drug is going to go to venous again. So we have this. Our role as a modeler is converting them at first to a schematic diagram that I can write the equations based on this. So I'm going to convert that system to this, which is for me more understandable as a mathematician. And with putting all of these rates from arterial, capillary, as you remember, I had some endothelial cells, this EC here, which are covering entire body vessels. They call it endothelial cells. And oops, sorry. And I put this compartment here. I have deep tissue. So overall, I'm going to write the equation for these 3 compartments here. So this drug or nanoparticles are coming from arterial, going to this capillary, and then endothelial cells, which are covering all of the blood vessels. On top of here, you can see all of this blood vessel, the walls are covered by endothelial cells. I ignored the effect of the interaction between endothelial cells and like surrounding cells. So we don't have any interaction here. And you can make this model definitely more realistic by adding other compartments, but I just kept it very easy for this mentoring case that I was doing. So, and we have a deep tissue. Using this schematic diagram, you write the system of equation, very simple, as you see, with very simple system of equations. And then when you use this model and the data that you have, we had some data, and you see that, for example, my model was really successful to fit the data that I had. All of this data using, the experiment that we had. And it was like the best model that we could find to catch the data. So as you see, this is the first like purpose for this project, finding a model that can catch the data. And then I need to find also the concentration of, we are looking for concentration of drug in the tissue, which we can, using this modeling approach, we can find this concentration. And the other question that they ask us as a mathematician or as a modeler, we should find we should approximate all of these parameters. So using this modeling approach, mathematical modeling, you can do that. There are many people who they don't know this, like mathematical modeling. They use some software to do that, but when mathematics is going to give us really more, I mean, comprehensive, more exact, I mean, results that people, that's why they trust us and ask us to do that. So, and there are some other statistical analysis that we did on some of the data. For example, as I said, when we have the model, we need to test it. The right-hand side, I had this data, in the left-hand side for this project, some other data for different drug, and we need to test the model if really this model is gonna really like follow the normality assumption or all of the assumptions that we have for a model that we use. So these are like some statistical analysis that we can do, people who are looking for. So, but since I'm going to go over the other like modeling approach, so I'm going to make it just shorter to start. This modeling approach, which is very interesting, actually it's very hard. I remember when I was working on the model, it was like 4 or 5 pages model, like it was really long, because now we are going to see what is going to happen for a drug when it's go to inside of the body, and it's going to get absorbed by different like organs. Like we have here, for example, I have 6 different compartments. So definitely the model is very complicated, but again, the approach is the same. This is compartmental modeling. So they call it, like in pharmacology or this area like physiology, they call it PBPK modeling. Let's go and talk about this type approach modeling. PBPK modeling actually is a good, very, I mean, useful tool that people, this is a very recent actually work, people in drug development, they use it. Of course, it was people that were using like software to do that, and we could develop a MATLAB package, which MATLAB is good because it's something that is a very strong and for computational like software, like we use it. And this package is going to help really to understand what is going to happen for this drug in different compartments, right? And actually modeling is very hard, and we cannot find a solution like analytically. So again, the goal is the same. We want to find the concentration of drug in different compartments that we have, and we are going to also approximate different parameters value that we have in the system. I put some part of equations, like not like 6 pages model. It's going to be boring actually, but the idea is the same. How we wrote the model for SIR modeling, or this 3 compartmental modeling, we do the same thing for here, but we need to assume there are some interaction between some compartments like kidney with other compartments, or we should include all of them. And this is the place that mathematicians, they cannot do it alone. So we need the help from people in this area. This modeling actually, the track drug movement entire body. So we are tracking. And it's good, as I said in the beginning of my talk, that this modeling, our goal is we give them a model, then they don't need to do further like animal experiments. It's going to be very good based on like, we don't want to sacrifice animal, or like it's going to be economically very better. So this is the goal for this, for example, project that we had data for some animal, And the goal was finding a model, again, PBPK model for a specific drug, which is oxytetracycline, for example, in swine. And it was the first time that we developed, so it was based on novelty and significance. It was really, and this actually oxytetracycline or OTC, this is very important in, for example, pork production. And it was feasible. We had like enough data, and we checked the data, and we provided the model, and we tested actually the model. It was a good model. So what is the model structure? I put this, I like the color. That's why I use a lot of colors in my modeling approach. Students, they like it actually. So I assume that I have this model for entire body. I have venous, I have arterial. You remember from this, that easy 3-compartmental model. So I have like this arterial and venous, and I have like 6 different compartments. I have muscle, lung, fat, like some other tissues here. I have kidney, liver, and as you see, there is an interaction between liver and stomach, and like intestine, and all of this. These are going to be like a very big, large, like model set of equations. Again, this is the ordinary differential equation approach, which is a model is very, I mean, if we know some biological, like if you have a good biological background, we can write the equation for this entire body. As I say, it was like 5-6 pages, but as you see also, we assume different like route of drug administration. Maybe it's injected through venous or intramuscular or through food or water. So this is this type, for example, drug, we assume all of this route. And so we study a 6-compartmental model for different liver, kidney, muscle, fat, lung, and plasma. And we added this new routes of drug administration for feed and water. So it was a new model. And I put just some equation just to show that this is not very hard. Of course, this is 6 pages, but the equations, as you see, this is they are just simple ODE model. And if I have, as you see, I have system of equation for each different compartments. For, like you see from the beginning, let me start from here, from liver, for blood compartment, for kidney, for muscle, lung, and fat. And this is the result for fitting with data. Of course, we didn't have enough data. The model, we need to check with more data. So not having enough data is actually a problem. Our people who are doing modeling, they have always this type of problem. So our model was for some of the, for example, for intravascular injection was okay, or for example, if we had intramuscular injection, it was okay. We didn't, as you see, I had data for kidney, for muscle, for liver. So it was not bad, but for example, for feed, I didn't have many data points. So it's not going to work here. Or for example, I need to have more data to really to check my model, to optimize my parameters and make it really a model that can be used for future prediction. This is the goal. And yeah, again, the result for again sensitivity analysis, this is what we are looking for, to see the effect of each parameters. I had like 32 parameters for each different compartments. I ran the same analysis for like global sensitivity analysis, changing all of the parameters at the same time, and see the effect of each parameters positively or negatively. They are going to affect, for example, this is the concentration of drug in kidney, liver, lung, or as you see, some of them, they have like they are not very influential on the model output. Some of them negatively, some of them positively. Fat, and so this is the muscle, plasma. So for each compartment, using this approach, LHS global sensitivity analysis, we can find the most influential parameters, which is going to affect the model output. So thank you so much for your attention. I try to not being very fast. Let me maybe stop share if any comment, any. So these are very, I would say, when I was mentoring my students, she really liked and she could learn a lot. I was happy with that one. There's a new
thing. Thank you very much. It's
Thank you so much.
Really nice work.
Thank you very much. Thank you.
Any questions from anyone?
I'll ask something really quickly. This was fun, really fun to watch, and I'll introduce myself. I'm the biology side of this equation, so it was really fun to hear you say the times you needed us. That was great. Just a thing.
Yes, always I say that your mathematics is everywhere. I love mathematics. And
So could I missed at the very beginning, what was this, a graduate student that you were working with, or is this a group of undergraduates that you were working with?
it was a specific one. I had one undergraduate student actually. I was working with her, and she was interested. She wanted to go for an internship, so she needed some background in this area for the internship. So
Great, yeah. So
yeah.
seems very feasible. I think it just, using compartmental models, series of differential equations, and then building up from 1 to 2.
Exactly. Yeah. Very simple.
Fantastic tool.
Yes.
Yeah.
Exactly. Yeah.
Yeah. It's great.
Thank you so much. It's good.
Hey.
The students, they have this chance to like do some projects, really, especially in math. The students, they are not getting like involved in many projects. So it's good they have this experience.
That's pretty sophisticated model for students. That's really good. I have maybe a little bit naive question related to one of your slides. I remember your pharmacokinetics model. There was I can't remember the details, but you said your model matched with data pretty nicely where you used ordinary differential equation. But I remember I see a big steep go down of one of the solutions. So it's not intuitive for me why there's a sudden change of it's not even a continuous function. I mean, it's continuous, but it's not a differentiable function at some places. So any extra comments about why is that?
Is it the first model, right?
It's either the first or the second. Yeah, it's one of the earlier ones. Yes.
Yeah, we had some data point which we where we discussed with the people who were like experts in this area, because we give them the model, and we had really some data points that were going up. It was like, I would say, I don't know if they are outlier or not. Our model could catch the data, average data, like average behavior, but there were really some point, I don't know, these points that are really based on the experimental, or we call it like maybe some outlier, or that the model couldn't catch them. As the model go up and then going, because as you say, this is continuously is going to catch the data.
Yeah, but I think you mentioned it actually catches the behavior. So your curve was going up and then drop dramatically. So from only the ODE point of view, like how did you come up with that? How did you get that solution? There has to be something changed, right?
Oh, for the second part of the model, right? Actually, I solved the equation for this project. I solved it analytically, and this is exactly analytic solution for the system, like 3 equations. And this analytic solution could show actually this is a real behavior of a drug, like going distribution and then elimination of drug. It was like the analytic
so maybe it's not as dramatic. Maybe the curve hide behind the dots. So I thought it's not differentiable. So maybe it's not that bad.
Yeah.
Sorry, you were saying?
Sorry, I mean, I was thinking that just we look at like this statistical test result, like R square, or it was, for example, for them it was, I mean, because we give them the model, if they accept the model or not, they know they are expert. I was just doing the math for the modeling part, but they need to say if really this is realistic or not. But it was really what they expected. For example, based on what is going to happen for a drug inside of one organ going up, and then elimination of drug, and then we have some exponential elimination of the drug, which they were happy. Yeah.
So I don't know if other people have question, but I have actually another question. I'm going to teach our undergraduate capstone course, which is mathematical modeling, in the coming year. So this is something I think is really nice and sophisticated enough for them to be a semester-long project. So I'm wondering if there's any further reading or your paper or your students' paper, or even such as like how can I find data, any extra resource that you can maybe point a direction.
Yes, yeah, actually for data is even I couldn't, for example, I was interested to make the model more complicated, but we need data. For example, you can maybe add more compartments, right? But if you just search for compartmental modeling, there are so many papers, and very easy that they just easily explain everything about how we write the model equation. For example, I didn't know really this approach. Definitely, I couldn't. I mean, when we look at the system equations, when I look at it, wow, I didn't expect really for a dry distribution, I can write this system of equation easily. But there's so many, like I would say, like literatures. If you just Google them, you can easily find just they just start from scratch to just explain each step how you write the model equations. And for data, no, unfortunately. I mean, I was lucky I could, there was an institute, they were working on animal data, like experiments, they were doing experiments, so they had data, but finding data is like, in this area, at least I know, I mean, I couldn't find, because even I wanted to make the model more realistic, make it harder, because as I said, we have some interaction between the cells and endothelial cells. I'm not expert, but I just ignored all of them. I just wrote a very simple equation. We have experts here and they know better than me. I just ignored and just wrote down the equations, a model, and when it catch the data, and when I test the model, and when I see that the result is okay, and they're happy with this result, it was like enough. And but for like making the harder, finding data is hard. Like we don't have public open access data in this, I know, in this but it's very nice, like compartmental modeling, because especially for this disease modeling, this is really good. The students start from first very simple, and step by step how they can write for really, we have very harder like type of disease. I am not familiar with, for example, how, for example, for cold or flu, what is the difference between them? Definitely maybe more, but this is very nice actually, and the students, they will like it.
Yeah. I'll just jump in. I'll toot our own horn a little bit. On Sunday, Becky Sanf and I will share a course that we teach, which is sort of a combination of biology and mathematics. We worked really hard to develop, first of all, the stepwise lessons, and we're targeting sophomores and juniors, so not necessarily a ton of math. As far as finding data, I would highly encourage you to find a biologist, or say what you want. Sometimes I could help you. There are data out there. There are data that are quite accessible. I spent quite a bit of time, Becky, I find, she'll tell me what math she wants to do, and I'll try to find the data sets that are published. And so a lot more data is also available in the public forum. So you can actually get raw data, sometimes depending, not always, depends on the funding of the experiment. So if you've got a project in mind, we can brainstorm about it, or chat with your local bio people, because very often they're sitting on data that they don't know what to do with. And that can be a real resource as well. Yeah, so I encourage that. And I'm going to run to my department meeting, but thank you for sharing.
Thank you so much.
Fun to see everyone.
Thank you so much for your comment. Thank you.
Thank you so much. So anyone else have questions or comments before we call it today? I mean, we're getting very close to the end anyway. And it's Friday, we don't have to push too hard. Okay. Well, I guess that's it. Thank you so much. I really enjoyed your
very
talk.
much.
It
Thank you so
was
much for
really
organizing.
nicely designed.
Thank you.
Okay, I will see you around in the next 2 days.
Yeah, see you.
Bye.
Bye. Take care. Bye.